Hello everyone, I am Nishmitha Gretta de Souza, working as an assistant professor in the Department of Pharmaceutical Chemistry at Yenepoya Pharmacy College and Research Center, Yenepoya deemed to be University, Mangaluru. Welcome to today's lecture on local anesthetics. So these are the contents that I will be covering in this video lecture. This session will cover the introduction, the history, the classification, mechanism of action and SAR of local anesthetics. By the end of this lecture, you should have a comprehensive understanding on how local anesthetics will work and their importance in medical field. So the SLOs of this video lecture are: students should know the definition of local anesthetics, students should understand the mechanism of action of local anesthetics, and they should understand the SAR, that is the structure activity relationship of local anesthetics. So, coming to the definition of local anesthetics. What are local anesthetics? Local anesthetics are drugs that can cause reversible loss of sensation in a specific area of the body without affecting the consciousness. So, they are commonly used in minor surgical procedure, dental work and in pain management. So, the local anesthetics are the drugs that can temporarily numb a small area of the body, allowing the patient to stay awake while the area is being treated. So, they can be injected or applied to the skin to reduce the pain during medical, surgical or dental procedures. Local anesthetics can also be used to treat painful condition, relieve pain after a surgery or prevent pain during an operation. Some local anesthetics are available over the counter and can help with pain, irritation and itching from minor cuts, sunburn and cold sores. Coming to the history of local anesthetics. The first local anesthetic was discovered was cocaine, which was derived from the coca plant. So, early use of these coca leaves, indigenous people in South America chewed coca leaves for their stimulating and numbing effect. Discovery of cocaine. The cocaine, the first local anesthetic, was isolated from the leaves by Albert Niemann in the year 1860. Its anesthetic properties were recognized in the later 1880s. Cocaine in medicine. In the year 1884, Karl Kohler, an Austrian ophthalmologist, introduced cocaine as a local anesthetic in eye surgery, demonstrating its clinical potential. Development of synthetic alternatives. Due to the addictive nature and toxicity of cocaine, the search for safer alternatives began. In the year 1905, Alfred Einhorn synthesized Procaine, which is marketed as Novocaine, the first synthetic local anesthetic, which became widely used due to its safety profile compared to the cocaine. Introduction of lidocaine. In the year 1943, Swedish chemist Niels Lofgren synthesized lidocaine, which is the first amide type local anesthetic. It was marketed as xylocaine and offered advantages like longer duration of action and less allergic reaction compared to the ester type anesthetics. Advances in the amide anesthetics. The development of more amide anesthetics followed, including bupivacaine in the year 1950s, known for its long-lasting effects, and ropivacaine in the year 1990s, designed to have a safer profile regarding cardiac toxicity. So, coming to the modern use and the research for these local anesthetic, today the local anesthetics are essential in various medical procedures. There are ongoing researches which focus on improving the efficacy, safety and reducing the side effect of the new drugs. Coming to the classification of local anesthetics, local anesthetics can be classified based on their chemical structure into two main categories. The first one is the amino esters. So, these amino esters are developed from the cocaine molecule, and these are considered as derivatives of benzoic acid. The amino esters are rapidly hydrolyzed in the plasma by the enzyme esterases. Few examples under amino ester local anesthetics are procaine, chloroprocaine and tetracaine. The next one is amino amides. The amino amides are developed from isogramin, which are considered as derivatives of aniline. So these are metabolized by the liver by the cytochrome P450 enzymes, and few examples under amino amides local anesthetic include lidocaine, bupivacaine and ropivacaine. So coming to the mechanism of action, local anesthetics work by blocking the sodium channels in nerve cells, preventing the initiation and propagation of action potential. The blockade inhibits the transmission of pain signals from the affected area to the brain, which causes the affected area to feel numb. The numbing effect can take place within minutes and last for a few hours. Local anesthetics will preferentially bind to the intracellular portion of voltage-gated sodium channels and inactivate it, leading to locking them in an inactivate state. This prevents the sodium ions from entering the cell, which prolongs the blocking effect and produces the desired clinical result. The ionization rate of the local anesthetic is a key factor in determining how quickly it starts working. And local anesthetics with lower pKa like lidocaine can act faster. There are several other factors which determine the action of local anesthetic. Few of them include the protein binding of the molecule and the metabolism of molecule. Local anesthetics can be administered by number of ways, which include topically, subcutaneously, intravenously, epidurally and intrathecally. Coming to the SAR, which is the important factor which helps us in designing the new drug molecule. So the SAR of the local anesthetic can be studied on three ways, that is focusing on the three main targets of the drug. The first one is the lipophilic center, which basically consists of the aromatic ring. So this provides the lipophilicity and helps the drug to penetrate the lipid-rich nerve membrane. Next will be the intermediate linkage. This linkage will determine what class of the drug is used. Either is it a ester or a amide drug is used as a local anesthetic. And this will also determine the metabolic pathway of the local anesthetic. And the last one is the hydrophilic center. So this includes the amino alkyl group. This provides us like how well the drug has the water solubility and allows for the formulation as a hydrochloride salt, which will determine its onset of action. So let us look into the SAR in detail considering one parameter. So moving on to the first one, that is the lipophilic center, which contains the aromatic ring. So the presence of the aromatic ring increases the lipophilicity of the molecule, so enhancing its ability to cross the membranes, that is basically the nerve membrane. Substitution on the ring can influence the potency and the duration of action due to its direct contact with the local anesthetic binding on to the sodium channel. The lipophilic substituent with the electron donating group in the para position of the aromatic ring of the ester type local anesthetic increases the anesthetic activity. So presence of an electron donating group like alkoxy, amino, alkyl amino on the aromatic ring will create a resonance effect between the carbon and the carbonyl group and their ring that results in the shift of the electron from the ring to the carbon oxygen. So as the electronic cloud around the oxygen is increased, the affinity of the molecule with the receptor also increases. Let us see few examples how the substitution at the para position increases the local anesthetic effect. The first example what we are seeing is a procaine. So procaine is an example for amino substitution in the aromatic ring. So you can see a aromatic ring, that is the benzene ring. At the para position there is a NH2 group, that is the amino group. So it is most commonly used in dental procedures to numb the area around the tooth, and is also used to reduce the pain in the intramuscular injection of penicillin. The next example what we are seeing is propoxycane. So propoxycane is an example for alkoxy substitution. So introduction of a propoxy group at the ortho position makes the ester hydrolysis difficult compared to the procaine molecule. So you can see that apart from the linkage, at the para position we have a amino group, and just next, that is from the linkage point, the very next carbon atom on the aromatic ring, that is the ortho carbon atom, you have a propoxy molecule that is attached. So this propoxy molecule will make this propoxycane a better compound when compared to the procaine molecule. So this propoxycane has got a longer duration of action, and it has been combined with procaine to accelerate its onset of action and provide longer lasting anesthetic effect, because procaine has a faster onset of action, whereas propoxycane will be used for a longer duration of action. So combination of propoxycane and procaine is proved to be very efficient. This drug is primarily used during dental procedures. The next drug what you can see is a tetracaine. So tetracaine is an example for alkyl amine substitution. So the presence of alkyl homologous series increase the partition coefficient in the increasing number of the carbon atom. So on the para position substitution, that is the amino group, one of the hydrogen group is substituted with the alkyl molecule. So over here in the tetracaine, it is a butane, that is a 4 carbon atom molecule which is being substituted. So this particular substitution, as the substitution increases, that is the maximum activity is shown by C4, C5 or C6 substitution. But in this example, that is tetracaine, you can see a C4 substitution in the molecule. So this tetracaine is most common used today in the topical ophthalmic anesthetic for short procedure on the surface of the eye as well as in the ears and the nose. When the aromatic ring is substituted with the electron withdrawing group like nitro or chloro, the electron cloud and the carbonyl oxygen is found to decrease, and as a result of this, the anesthetic activity also decreases. Introduction of electron withdrawing chloro group in the ortho position of the procaine result in the compound that is called as chlorprocaine or chloroprocaine. So this chlorprocaine makes the ester bond more susceptible to hydrolysis. Therefore, chlorprocaine has shorter duration of action, and it is thought to have the lowest CNS as well as cardiovascular toxicity of the agents in current use. So this chlorprocaine is commonly used for epidural anesthesia. Coming to the next part, that is the intermediate linkage. The linker is usually a ester or a amide group along with the hydrophobic chain of varying length. So when the number of carbon atoms in the linker is increased, the lipid solubility, protein binding and the duration of action as well as the toxicity also increases. So you can see on this slide there are two types, one is the ester type and the amide type. So the linker or the intermediate chain will decide whether the local anesthetic is categorized as a ester type or the amide type. In the same category, you can see a R substitution that is attached. So this R substitution is nothing but the hydrophobic chain. So as the hydrophobic chain increases, the lipid solubility, protein binding, duration of action and toxicity also increases. So esters and amides are basically bioesters. So that is, they have a similar size, shape and electronic structure and also have a similar binding properties. They only differ in their stability in vivo as well as in vitro. For the drugs that only differ at the linker functional group, amides are more stable than esters and thus amides have longer half-lives than esters. The nature of substituents on the aromatic ring, which we already studied earlier, can affect the electronic nature of this linker molecule and can contribute to the drug of potency, higher potency and stability. So they also give a steric block to protect the linker from metabolism. So the binding affinity and stability of the anesthetic molecule is affected by the linker and the functional group on the aromatic ring. In general, ester molecules are more susceptible to hydrolysis than the amide functional group. Because of this nature, the amides are preferred over esterases as esterases are widely found in the blood and liver. Coming to the last part, that is the hydrophilic center, which basically includes the amino alkyl group. So this amino alkyl group is essential for the drugs to improve the solubility in the water and to form a stable salt. So it also influences the pK of the molecule and affecting the onset of action. The amino group that is attached is either a tertiary or a secondary amine. So the tertiary amine are more preferred as they are less irritating to the tissue. Substitution with more number of carbon atoms increases the partition coefficient and thus increases the activity of the local anesthetic. So here are few of the example wherein you have procaine, benzocaine, lidocaine and tetracaine. So compared to the most of the molecule, procaine, lidocaine and tetracaine are all tertiary substituted molecules. So lidocaine and procaine have got higher substitution on the tertiary amine, that is, it has a C2H5 substitution which are comparatively better to benzocaine and lidocaine. The nerve membranes are generally lipoidal in nature. An increase in the lipophilic character enhances the penetration of local anesthetic into the membrane but decreases the solubility of the drug into the extracellular and intracellular fluid. An increase in the hydrophilic portion of the local anesthetic decreases the drug's penetration into the membrane. Hence, the local anesthetic should have equal balance of both lipophilic and hydrophilic center. Binding of local anesthetic to plasma and tissue protein is due to the presence of van der Waals forces, dipole-dipole interaction and electrostatic force of attractions. Compounds having high potency and long duration of action tend to bind more strongly to the plasma and tissue protein. Different enantiomers of local anesthetic exhibit differences in their terms of their selectivity and toxicity, as well as they differ in their duration of action of local anesthetic, and also it affects the stability of the compound. Coming to the summarization of the whole lecture. So, local anesthetics are vital in medical practice for the pain management and minor surgical procedure. The classification, mechanism of action and structure activity relationship are the key in understanding as well as designing the new compound having optimum activity. So, understanding the SAR of local anesthetics will help in designing a drugs with specific properties like onset of action, which is determined by the drug's pKa as well as lipophilicity, then duration of action, which is influenced by the metabolic pathway and protein binding, and finally the potency, which is affected by the lipophilicity and the ability to bind to the sodium channels. So, local anesthetics are vital in medical practice for pain management and the minor surgical procedure. Thank you for joining this lecture. These are few of the references which were used to prepare this particular presentation. Thank you.